Okay, we're going to welcome everybody and to our chemistry seminar series. So we're glad all of our students are here and those maybe they're watching online and our speaker who's joining us online. So we're very thankful to have Dr. Is it Lavis or Lavis?
It's Lavis.
Lavis. You know, as a Midwesterner, we have a hard time with A's. So
yeah,
I am from well, so thank you, Dr. Lavis, for joining us. And I will introduce him in just a moment, but we want to just recognize once again that this is our 75th anniversary as a department. So many people don't know about Andrews University, but we have been around for a while. And then it's also the 50th anniversary of our seminar series. So we're glad to have a prominent young scientist. And young, of course, means less than 40, I think. Is that what that title meant? I don't. He got Dr. Lavis has an award for being one of the top 12 scientists here. That was recognized, I think, was it by the American Chemical Society or Chemical Engineering? So, so I'm going to go ahead and read the abstract for his talk today. It was well written and it's very nice. So no sense of reinventing the wheel. And then we'll turn the time over to our speaker. So Luke Lavis is a group leader at the Howard Hughes Medical Institute in Genelia Research Campus in Ashburn, Virginia, which for those joining us online and who haven't heard, they're going to get a lot of snow tonight. So they get to experience the solid phase of water in great quantities. He received his bachelor's degree at Oregon State University in 2000. And he performed undergraduate research in synthetic organic chemistry with Professor James White. He received his PhD in organic chemistry in 2008 from University of Wisconsin-Madison under Professor Ronald Rains, developing strategies to track anti-cancer proteins in living cells. So he was born and raised in Applegate, Oregon, and he worked in the biotech industry after completing his bachelor's degree. He first worked at Molecular Probes in Eugene, Oregon, which I got to visit when I was in industry. And then he went on to Molecular Devices down in California. And then with these work experiences, that convinced him to go into research rather than medicine. And we have a number of students. Yeah. So we can relate to that here. That's one of those ongoing struggles that our students are facing, and then the faculty have faced as well. So now, though, he's in Genelia, and he works at the chemistry-biology interface, developing fluorescent dye labels for single molecular imaging, targeted molecular delivery, and probes to map cellular activity. So we get to hear more about that. And as I was just mentioning, he was recognized by the Chemical Engineering News last year as one of the talented 12. It sounds like a team of superheroes, and perhaps it is. And his superhero name is the fluorescent phenom. And so we welcome the fluorescent phenom to Andrews University, and the floor, well, actually the screen is yours.
All right. Well, thank you very much for the introduction. Hopefully everyone can hear me. I apologize for my voice. I'm a little under the weather, but hopefully you'll be able to understand what I have to tell you today. So I'm going to tell you a little story about taking old school dyes, molecules that we've known in some cases for over a century, and using modern synthetic organic chemistry to transform these privileged scaffolds into tools for 21st century biochemistry and biology. And I've kind of given away the punchline on the title slide. Basically, what we've discovered is by replacing dimethyl amino groups with this little four-membered ring, this azetidine ring, in a wide variety of fluorophores, we can greatly improve the brightness and the photostability of these dyes without affecting the structure much. It's just the addition of a carbon atom or two. And today I'll tell you how we got here and kind of give you some examples of these molecules in action. First, though, I'll tell you where we do our work, and that's the Genelia Research Campus. And Genelia is basically an experiment by the Howard Hughes Medical Institute. The Howard Hughes Medical Institute was founded by Howard Hughes, who was at the time the richest man in the world. And he gave a large portion of his fortune to fund medical research. And for many years, HHMI would fund researchers around the country at different host institutions. But about 15 years ago, they decided to build their own research campus, and Janelia was the result. So here's Janelia, here's the campus. We're located in Northern Virginia, outside Washington, D.C., and we're fairly young. We just opened in 2006. And so the idea was to bring a bunch of scientists from different disciplines together, give them, let's call it more than adequate funding, and then turn them loose on a couple big problems. And so we're now the home to around 45 or so group leaders. There's no teaching, there's no tenure, and the lab size is small. So you start out with 2 people and you can grow to as large as 6, but there's no real empire building at Janelia. We're fully funded by HHMI. We're not even allowed to write for external grants. And the 2 main research aims are developing new imaging modalities and cracking neural circuits, so imaging and neuroscience. And my lab is the only organic chemistry lab at Janelia, which basically means I get to hang out with a bunch of crazy, awesome biologists and optical physicists who have really interesting problems. And in some cases, these problems are even scientific ones. And so we get to go back to the lab and use organic chemistry principles to design and build small molecules that can help solve these problems. And the main type of molecule that we work on are fluorescent dyes. And in this plot, I show 30 commonly used fluorescent dyes in biology. They're plotted according to their brightness, which can be defined as their extinction coefficient. That's the absorptivity of the molecule, times their quantum yield. That's how many photons you get out over how many photons you get in. And that's plotted against the absorption maximum of the dye. That's what each one of these points denote. And then the corresponding structures are colored according to the emission maximum of the dye. Now, the point of this slide is not to go through every structure, but it's just to showcase that over the last 100, 150 years, chemists have discovered a large number of fluorophore scaffolds. And now we can take these molecules and tune or tweak them for particular biological applications. But before I get into some of our work, what I wanted to do is just answer maybe a question that you might have, and that's, where the heck did we get these fluorophores? Where did they come from? What's the story behind their discovery? Because it's an interesting story, and it'll put some of our work on developing new chemistry in context. And the story of fluorescent dyes and the discovery of fluorescence is intertwined with 2 other stories. The first is kind of obvious. That's the discovery of synthetic dyes in the mid to late 1800s. And the other intertwined story is not so obvious. It's actually malaria, which was caused by this little plasmodium parasite. Of course, that is transmitted by mosquitoes. And this is a false electron micrograph of a juvenile plasmodium inside the gut of a mosquito. And so I'll tell you kind of how malaria plays a big role over almost 100 years in the discovery of fluorescence, the discovery of synthetic dyes, and the development of fluorescence instrumentation. And the reason malaria started out as sort of the impetus for the discovery of fluorescence is a small molecule called quinine. Quinine is an effective anti-malarial treatment. It's been used by indigenous peoples for many years. It's a natural product from the bark of cinchona trees, and it probably acts by inhibiting hemozoin. So as your red blood cells are being eaten up by these parasites, they encounter a lot of heme, and that's toxic to the parasite, so it has to sequester it in some way, so it biomineralizes it into this hemozoin. And quinine and other similar drugs are thought to act by breaking up these crystals that leads to free heme and toxicity. And if you're a European power in the 1600s and on, and you're trying to get a foothold in different areas of the world where malaria is rampant, you might think, let's put quinine in the water, and that's what they did, and that's where we have tonic water. So it's still used in tonic water today. It gives that nice bitter flavor. And quinine was actually the first small molecule fluorophore, and the fluorescence was first observed by John Herschel, who was a really jack of all trades, master of many, fantastic scientist in several different areas. And he published this paper in 1845, and it's titled, though they wrote papers differently back then, and it's titled "On a Case of Superficial Color Presented by Homogeneous Liquid, Initially Colorless." And basically, the take-home message of this fairly long paper is that he put a bunch of quinine sulfate, which was the stuff you could buy, in water and put it out in the sun, and he saw this weird blue color coming off of the top, and that was fluorescence, and he just noted that and then went on to do other things. And a few years later, George Stokes actually worked out the process of fluorescence, again using quinine, in this wonderful paper entitled "On the Change of the Refrangibility of Light." And in this paper, he details a number of experiments and realizes that he has found a new phenomenon. And so he called this phenomenon initially dispersive reflection. And so if this name had stuck, we would still be calling, fluorophores, we'd be calling them dispersive reflexiphores, and we'd be using dispersive reflexometers or something. But even in this first paper, Stokes noted, and he said, in a footnote, "I confess I do not like this term. I am almost inclined to coin a word and call the appearance fluorescence." And that's, of course, the name that stuck. And the way that Stokes figured out that fluorescence wasn't just some weird scattering of light was an experiment like this. And this is the legend. Whether or not this actually happened is up for debate. But the idea is that Stokes took a bottle of quinine and went into a church and used the blue stained glass window as a filter to only transmit blue light. This would be absorbed by the quinine, and then the molecule would then emit a longer wavelength of light. And then he used a yellow glass of wine as the emission filter. And they could observe that he wasn't seeing the blue light just being scattered, but actually new light, new photons that were longer wavelength. And in his honor, we call the difference in the absorptivity, the absorption maximum and the emission maximum of fluorophores, the Stokes shift. So fluorescence sort of was this interesting phenomenon that had been discovered. And around the same time, the Industrial Revolution was in full swing. And because of that, there was this stuff called coal tar laying around. It was widely available. And it's a byproduct of coke and natural gas production. And so the lamps that were lighting the streets of London at the time were also resulting in a large amount of this coal tar. And of course, it's a complex mixture of organic compounds. And it turns out to be a nice source of starting materials for organic synthesis. And so while we might think of coal tar as this dirty thing from sort of an environmental standpoint, at the time, chemists realized that you could distill out really interesting molecules and then do new chemistry on these purified molecules. So they called this type of chemistry synthesis. And the main proponent of this type of chemistry was von Hofmann, who was the president of the Royal College of Chemistry in London. And von Hofmann had a student in his lab, Willam Perkin. And together they decided to try and make quinine. Because they thought, why should we try and grow it in a plantation half a world away when we could make it from coal tar in the lab? And so this was the attempted quinine synthesis. They were going to take N-allyl-toluidine and oxidize it to give quinine. Now, everyone in the room will probably laugh at this because it's a little bit silly. There's no way this would ever work. But remember, at the time, they didn't have NMR or any other really types of spectroscopy to get at molecular structure. All they had was elemental analysis, so they could get the chemical formula. And when you look at the chemical formula, it kind of makes sense. If you take this molecule, you have the right number of carbons and nitrogens, you add some oxygen and take away water and fuse two of these things together, well, everything works out. So they tried this, and of course it didn't work. But for some reason, Perkin decided to keep trying to oxidize aniline compounds. And so he had built a little lab in his parents' attic. And so he distilled out aniline, and it was kind of dirty, so he got these other toluidines, and he oxidized them. And from the goopy mixture that he got, he found a purple compound, which he called movine. And purple was a big deal because purple colored compounds were always associated with riches and royalty. The best purple dye was Tyrian purple, this brominated indigo derivative, and it was found in these spiny diamuric snails in very limited quantities. And so only the richest people could afford a purple robe. There was also poor man's purple, which could be generated by fermenting lichen, but this washed out rather quickly, this orcine molecule. And so what Perkin realized is that he had developed a new synthetic purple molecule that he could make very easily from stuff that he could distill from coal tar. And so he promptly dropped out of graduate school, filed a patent, and started a company making aniline purple or movine, as well as researching other dyes. And this first compound, this discovery of synthetic colored molecules, is really thought to be the start of the modern chemical industry. And so there was a flurry of activity in the late 1800s, leading to a large number of molecules, some of which we still use today. So methylene blue, Comasi brilliant blue, which is used to stain proteins in gels. Ponce-OS used similarly. Indigo was also synthesized in a lab rather than being grown in a field in India. And so the color of your blue jeans was probably made in a factory somewhere rather than extracted from a plant. Other naturally occurring colorants were also made in the lab. And fluorophores were also started to be made synthetically. And I'll talk a little bit more about fluorescein, first synthesized in 1871, a little bit later. All right, and the effect of malaria would have one more sort of kick in the pants for fluorescence technology, and that was because during World War II, Imperial Japan controlled a lot of the chinchona tree plantations. And so the US malaria program, which rivaled the Manhattan Project in terms of manpower, sought replacement anti-malarial drugs during World War II. And so the idea was, well, they knew quinine was fluorescent. They thought that many other drugs based on quinine would have this quinoline fluorophore moiety. And so they thought, well, maybe we could use a fluorometry to test concentrations of various drugs in the blood. And so they did sort of the first pharmacokinetics types experiments using fluorometry. And so this resulted in the improved dosing of atabrine. Atabrine was a known molecule at the time, but it didn't work very well for the soldiers. And what they realized is that it was taken up by a lot of the fatty tissue. And from these experiments, they realized they needed to give a very large dose initially, and then they could give smaller doses after the tissues were more or less saturated. And it also led to the discovery of a new antimalarial compound, chloroquine. And because of this effort, fluorometers, which were prior to World War II a real novelty in the lab, were commercialized, and now they're found in essentially all labs around the world. I'm sure you guys have fluorometers at Andrews. I heard you're going to learn about fluorometry next week. I think my lab has 3 or 4 fluorometers. So this really changed everything because it made them available to a wide variety of scientists. All right, so coming back to this chart of fluorophores, my lab works on kind of old dyes. So we focus on the xanthine fluorophores, which include fluorescein and the rhodamines. And these are near the top in terms of brightness, and basically span the visible region of the spectrum in terms of absorption. And they're also quite old. Fluorescein, as I mentioned before, was first synthesized in 1871 by Adolf von Baeyer, and the rhodamines were first synthesized about 10 years later, popping up in the patent literature in the 1880s. And rhodamines in particular are really fantastic scaffolds for fluorescent probes. You can tune their structure or their spectral properties through very simple substitutions. So here's rhodamine 110, the simplest rhodamine, which absorbs blue light and emits in this green color. If you alkylate these aniline nitrogens, you can shift the spectral properties 50 nanometers into the red. And if you further fuse these rings together, you can buy yourself another 25, 30 nanometers. Another way to modulate the spectral properties of rhodamine dyes is to replace this oxygen with a carbon or a silicon atom, and this buys you 60 or 100 nanometer shifts into the red. You can also make fluorogenic molecules out of rhodamine dyes, molecules that turn on their fluorescence when they interact with an enzyme or some other stimulus. And here, if you acylate these aniline nitrogens, instead of alkylate them, you can lock the molecule into this lactone form, where this extended conjugation, normally seen in rhodamine 110, is broken. And this gives a molecule that's colorless and non-fluorescent. It absorbs only in the UV. But if you remove these groups, in this case, removing of these orthonitrobenzyl cages with light, you can recapitulate the fluorescent structure, causing a large increase in both absorption and fluorescence. You can also alter this open-closed equilibrium by changing groups down here in a variety of different ways to make fluorogenic molecules. And one other thing that's often underappreciated is that this equilibrium between this closed lipophilic form and this open zwitterionic form is dynamic. And it makes the molecules sort of amphipathic, so they can exist in a lipophilic environment like a cellular membrane, but they're also highly water soluble. And this makes these sorts of molecules very nice for live cell labeling experiments because they can slip through cellular membranes and get washed in and out fairly rapidly. So those are the good things about rhodamine dyes. The problem for the last century has been that rhodamines are difficult. They use very archaic chemistry. And so here's the classic synthesis of rhodamine dyes still used today. You take an aminophenol and this phthalic anhydride, the carboxyl group here. You cook them together for a couple of days in strong acid, some harsh heating, and you generate this molecule, which is a mixture of isomers, which can be purified out to the desired molecule, in this case, 5-carboxy tetramethylrhodamine, using chromatography, typically HPLC. Now, the problems with this sort of synthesis are many. The nitrogen substitution is fixed. It's hard to change the nitrogen substitution after the rhodamine has been formed. You get fairly low yields of this molecule because this highly charged compound is difficult to isolate. And you get this isomeric mixture, which requires a tedious purification. So this is a bit of a problem. And if you look at the cost of these molecules, this is $1.72 a milligram. And this is an order of magnitude more expensive. So this chemistry is in contrast to a very similar molecule, fluorescein, which uses the same chemistry, but it's a little bit easier. Here you use resorcinol, and again, a phthalic anhydride. Slightly milder conditions can generate this molecule, which is easy to isolate because it's a neutral molecule at acidic pH. So you can just precipitate it out, dump it into water. And then you can isolate these isomerically pure compounds, not through chromatography, but by recrystallization. And this is reflected in the price. This is only 3 cents a milligram. And isomerically pure fluoresceins are only $1.38 a milligram. So we had a very simple idea a couple of years ago now, where we thought, okay, well, this guy looks like this guy. Can we somehow turn fluoresceins into rhodamines? And that was the idea. And we did it using a modern chemical reaction, a Buchwald-Hartwig cross-coupling reaction, where we would take a fluorescein, make the bis triplet, which you can do in just one step, and then we could couple on a variety of different amine nucleophiles to generate a diverse array of rhodamines. And the nice thing is if you have substituents on the fluorescein, you can get these substituents persist on the rhodamine dye. So that allows us to make a large number of rhodamine dyes. And so just as a start, we tried fluorescein. We made this bis triplet, and then an 85% yield, we could couple on a fairly volatile amine to generate tetramethylrhodamine. And over the last few years, we've explored the scope of this reaction. Again, taking just simple bis triplets and coupling on primary and secondary amines to make fluorescent rhodamines, amides to make directly fluorogenic enzyme substrates for protease enzymes or esterase enzymes. We can couple on anilines to make quencher dyes. We can couple on carbamates to make fluorogenic molecules, photoactivatable compounds. We can also take carbamates and use them as protected ammonia equivalents to make simple rhodamines like rhodamine 110. If we start with fluorinated fluoresceins, we can get fluorinated rhodamines, which are much more photostable. And then we can also use carbofluorescein to make carborhodamines, both fluorescent and fluorogenic compounds. And we can have also made the silicon-containing fluorescein to make these silorhodamines. So overall, we can make a large variety of rhodamine dyes. But one thing we wanted to do is try and improve, use this technique to improve labels for single molecule assays in live cells. And so this is a growing area of biology and biochemistry, the ability to look at individual molecules rather than averages of populations of molecules. And the idea inside live cells is to somehow tag a protein of interest with a bright photostable fluorophore, and then watch as it moves around, say, the nucleus of a cell. This could be something like a transcription factor or some other regulatory protein, and you can then correlate the dynamics of individual molecules with cell behavior or cell fate. And so the ability to localize and track individual molecules inside live cells has been really pushed forward by developments in microscopy. So this is a movie of individual transcription factors that are being tracked in 3 dimensions using a multifocal microscope, a microscope that takes images in 9 different focal planes simultaneously, so you can cover a large portion of the nucleus this way. The other bit of enabling tech has been a biochemistry technique that was first developed by Kai Johnson in Switzerland, and these are called the so-called self-labeling tags. And the idea here is you express your protein of interest as a fusion with one of these tags. They react specifically and irreversibly with a ligand motif that you can attach to a fluorophore. And that allows you to make a fluorescent bioconjugate with a synthetic fluorophore inside a living cell. And our favorite system is the halo tag system. So the protein part is based on a bacterial dehalogenase. It's basically got half the catalytic machinery taken out, so it reacts irreversibly with little ligand motifs. It's a little bit larger than the green fluorescent protein, and it's a commercial product from Promega. And the reason we like it is that the second order rate constants of this enzyme are off the charts. They exceed 10 to the 7th. The ligand motif is this short little chloroalkane. And the dye that was used in this experiment and many others, the really only commercial dye at the time that was bright enough to look at individual molecules was this tetramethylrhodamine, something I mentioned before. So tetramethylrhodamine, or TMR, is great. It gets into cells quite rapidly. It's small enough to react very quickly with the halo tag. But the problem is that its quantum yield is fairly low at 41%. So we set out and thought, let's design and build a better version of tetramethylrhodamine that would still be small, but simply be brighter. And so the first question we asked is, why does tetramethylrhodamine have such a low quantum yield? And indeed, it's a bit of an outlier. So rhodamine 110, 88% quantum yield. So 88% of the time it absorbs a photon, it'll emit another photon. That's excellent. Dimethyl rhodamine and N' dimethylrhodamine, 84%. Tetramethylrhodamine, 41%. So something's going on here. And when you think about the process of fluorescence, you have a molecule in the ground state, it absorbs a photon, this gives an excited state, and then the molecule can relax back down to the ground state by emitting another longer wavelength photon. But this only happens 41% of the time with tetramethylrhodamine. What about the other 59%? And a likely explanation for this is that TMR is particularly susceptible to non-radiative decay back down to the ground state through a twisted internal charge transfer state, or TIC state, where you get electron transfer from this nitrogen into the ring system and a twist around this carbon-nitrogen bond. This funky diradical intermediate can relax back down to the ground state without emitting a photon. And it's also a formal diradical that can undergo irreversible photobleaching reactions. So the idea is if we could raise the energy of this state, basically decrease flux through this pathway, we can improve quantum yield and perhaps photostability. When you think about the structural parameters that are important for promoting or inhibiting this ticked mechanism, you can think about the order of the carbon-nitrogen bond, how much double bond character there is, how easy is it to twist. The ionization potential of the nitrogen, how easy is it for the nitrogen to give up an electron. And then any homoallylic interactions between substituents ortho and alpha to the nitrogen, which might also predispose the molecule to twist. So we had a very simple idea, and we thought we could control all of these parameters simply by tying back the substituent into some sort of ring system. And this idea didn't come out of a vacuum. We and others had shown that changing ring size could affect brightness. So in previous work, we had made tetramethylrhodamine, this piperidine containing rhodamine, and this pyrrolidine containing rhodamine with 6-membered rings and 5-membered rings. And when you look at the properties, it was actually striking how this very small change in structure could affect brightness. And I'll just focus on the quantum yield values. But tetramethylrhodamine has a 41% quantum yield. The 6-membered ring actually goes down to only 10%, and the 5-membered ring goes up to 74%. So we initially made some ligands based on this pyrrolidine containing compound, and they weren't much better than tetramethylrhodamine in live cells. So we needed to go to something brighter. So we had this sort of crazy idea. Let's try an azetidine containing rhodamine, this 4-membered ring. And to be honest, I didn't think this was going to work, and no one else did either. I had future chemistry Nobel laureates saying, I don't know, Luke, that ring looks pretty strained. And the reason is pretty simple. All of you have had organic chemistry, and we know that 4-membered rings are strained. There's 90-degree angles, and we all know that carbon likes 109.5-degree angles, right? And it just didn't seem likely that the planar delocalized system would be compatible with this highly strained ring. And there was another problem, and one of the reasons that azetidine had never been explored in any fluorophore was that the chemistry that people typically use to make rhodamine dyes just wasn't compatible with this little ring. We tried this, it doesn't work because this ring under these very harsh acidic conditions pops open. Now, what changed our mind, we did a little bit of molecular modeling, and we were surprised, and you'll see this in a moment, that the predicted structure was planar. And that sort of piqued our interest. And when we dug down a little bit deeper, what we found is that of all of these dyes, the azetidine had the predicted shortest carbon-nitrogen bond length. It had the longest interatom distance, so because of these nice 90-degree angles. And it was also known that azetidines have a very high ionization potential. And so that led us to predict that maybe this would actually work. Maybe it would be brighter than tetramethylrhodamine. And then we had also fixed the chemistry problem. As I showed you earlier, we can take fluorescein-bis triplets and then use palladium-catalyzed cross-coupling reactions to isolate rhodamine dyes. And so this works in very nice yield. And so we went ahead and tested the spectral properties. And we saw a surprising increase, a 25% increase in absorption, and a more than doubling of the quantum yield to near 90%. And so it was evident from the moment we took this out of the HPLC, where we first purified it, that this dye was going to be a winner. It was just so bright, even to the naked eye. So we call this dye Janelia Fluor 549 because it was developed at Janelia. It's a fluorophore, and it absorbs at 549 nanometers in water. And just to remind you, it's about 2.5, 3 times brighter than tetramethylrhodamine. But we do this through a very small structural perturbation, just the addition of 2 carbon atoms net. So this preserves a lot of the nice properties that we like about rhodamine dyes. The spectra are basically the same, TMR and JF549. We can use our chemistry to quickly make JF549, in this case, halo tag ligands for this labeling technology from simple carboxyfluorescein starting materials. We can label live cells easily. The dye washes in and out very rapidly. These are cells expressing halo tag histone H2B fusions. If we label with lower amounts, we can resolve individual molecules and get statistics. And we find, compared to tetramethylrhodamine, that JF549 is twice as bright and also lasts twice as long. So that's overall a 4-fold improvement in photons per molecule. We can also do super resolution microscopy, localization microscopy, and under reducing conditions, the dyes switch on and off, and we can get these sorts of pictures. Again, this is halo tag histone H2B, and JF549 emits about twice as many photons per localization event as tetramethylrhodamine. And this has allowed us to do some really beautiful experiments in collaboration with Eric Betzik and Bob Tejan and James Liu here at Janelia, where we looked at where a transcription factor, SOX2, binds stably in the nucleus of an embryonic stem cell. And we found that it actually binds in these little enhancer clusters, and it was unknown whether it would bind more or less evenly throughout the nucleus or in these little clusters. And so we actually showed for the first time that indeed it binds in these little segregated areas. Now, I've shown you this works for rhodamine dyes, but there's a large number of classic fluorophores that contain these dimethylamino groups. And so we were curious, would this be a general solution to improve the brightness of fluorophores across the visible spectrum? So for rhodamine dyes, I showed you that we got a 2-fold improvement in quantum yield. For methylaminocoumarins, we got a 4-fold improvement in quantum yield. 3-carboxyaminocoumarins, we get a 28-fold improvement in quantum yield. Naphthalides go from non-fluorescent to highly fluorescent just by adding a carbon atom. Acridines, a 2.5-fold improvement in quantum yield. Rhodol is a 4-fold improvement in quantum yield. Carborhodamines, 30% improvement. Silicon rhodamines, a 40% improvement. And oxazines, a 3.5-fold improvement in quantum yield. So this is a really nice general strategy for taking fluorophores, which are found in a wide variety of probes, and just making them brighter and more photostable. And so we're slowly going back and retrofitting a lot of classic molecules and making them significantly brighter. And just one last thing we're doing on this sort of project is we found a way now to fine-tune the spectral and chemical properties just by putting substituents on the azetidine ring. And so we can tune the spectral properties by up to 25 nanometers. This goes really nicely according to the substituent field effect, or the sigma I Hammett constant. We do this without changing the extinction coefficient, the absorptivity, or the quantum yield, so everything remains high. We've also found that we can modulate, use these substitutions to modulate the open-closed equilibrium, and we do this by looking at the absorption of the dye in different dioxane and water combinations. And we find that as we go down to more electron withdrawing groups, the dye closes up in more modest dielectric constant environments. And so basically the take-home message is that there's a higher propensity of the molecule to close. And so I'll skip over this because I know we're running a little late on time, but we've been able to then use this information to rationally design a new fluorophore, JF585, that has really fantastic wavelengths matching common laser lines, but it's also fluorogenic, so it doesn't absorb until it binds its halo tag. And so you can get really beautiful images like this without having to wash out excess ligand. And so basically what we've discovered is a new generation of cell-permeable dyes. When I was at Molecular Probes, we worked on the Alexa fluor dyes. These are fantastic dyes for antibody labeling. I actually made the first gram of Alexa 633. That was my first project. But now we have the Genelia fluor dyes where we can match all of these nice spectral ranges, but have them much smaller and much more cell-permeable. And so I'll just end with a little story on super resolution imaging that we did with Eric Betzig here. And the idea is we can take a dye that binds to a particular cellular structure at a very small amount, and then watch individual molecules, localize them, image them until they bleach, and then use that information to build up a high resolution map of the cellular structure. And so we've screened through our large library of rhodamine dyes. We found one with these azapine rings that stains internal membranes. We've also engineered stains by taking our Genelia fluor dyes and attaching them to known DNA binders. These are fluorogenic far red DNA stains. And then using a really fantastic, awesome microscope developed by Eric Betzig's lab, we can then image molecules in 3D. And so we scan through, we only look at bound molecules. Here's an example of a HeLa cell during mitosis. We can do this sequentially. Here's an example of a cell where we're actually able to image not just nuclear DNA, but also mitochondrial DNA. And we can do this serially by looking at DNA first, and then nuclear lamins, and then internal membranes. And so I'll just show you a movie where we're going through a cell. Again, the purple is DNA, the yellow is the nuclear membrane, and then the blue is the internal membranes. And so you can see these large tubular structures, which are the mitochondria. You'll see mitochondrial DNA, these little purple spots, and this web-like structure is actually the endoplasmic reticulum. And so we're slowly now building up a larger collection of these paint stains for super resolution microscopy. And the hope is combining this with other types of super resolution microscopy, we might be able to generate, say, a sort of a prototypical cell with many different proteins and other cellular structures at resolution that's much better than you can achieve using normal diffraction limited imaging. All right, so I'll just end with some acknowledgements. So as I mentioned, my lab is small. Actually, Adam is also just moved back to Michigan. He's a Michigan native. Most of the work done was by John Grimm, who is a fantastic medicinal chemist that actually stole from Merck. We have fantastic collaborators on the imaging and biology side, as well as a number of outside collaborators. So I want to thank them for their hard work, you for your attention, and I'm happy to answer any questions. Hopefully I didn't go too long. Was that okay?
No, you did great. Thank you.
Make sure and give your name.
Hi, I'm Dylan. I'm curious with the success you had with the 4-membered rings. Have you thought about using other ring synthesis like an 8-membered ring? I know a 3-membered ring would be extremely strained, but has your lab looked into the synthesis of other types of rings?
Yeah, that's a great question. So I mentioned this at the end. This 7-membered ring compound was actually part of that series. We made this one as well, just for fun. We haven't gone to the 8. We've also made the 3-membered ring, the aziridine, but that's now so strained that the molecule is colorless and non-fluorescent. So the idea is now the nitrogen puckers, and then that causes this carboxyl group to bite and it interrupts the conjugation. So it's actually a really interesting compound because it also seems to interact with reactive oxygen species to form what we think is fluorescein. So we've thrown it at cells, it stains peroxisomes, but we haven't investigated it further. It's sort of a really interesting molecule, but unfortunately it's not that bright.
Hi, my name is Helen, and I also had a question about the four-membered rings. I was wondering what property of the four-membered ring makes it so that it consistently increases the fluorescence of the molecules that you were working with.
Mm-hmm. Yeah, so I'll go back to this slide. So it's probably a combination of things, but in this slide here, if you just have no methyl groups, 88%, one methyl group on each side, 84%, and then it goes down to 41%. So this, I think, has puzzled people for a long time because this general trend is seen in all different types of fluorophores. And so it's probably the most, the biggest cause is the ionization potential. So it's known that fully substituted anilines have a fairly low ionization potential. And so it's easy for these anilines to give up an electron. And so that charge transfer is favored with fully substituted aniline nitrogens. And so that's probably the main cause, but there's also these other things where if you strain the ring, for example, you'll get a shorter bond. That's known from sort of simple molecular orbital theory. And then also these interatom distances may also play a role, but it's probably mostly this ionization potential. And azidanines are well known to have an extremely high ionization potential.
Hello, my name is Alistair. I was wondering if you had any internship positions or student researching position. It's actually a very interesting field.
Yeah, so we have a Janelia undergraduate scholars program. I think it just ended. I don't know. But that's the main mechanism. We just have a 10-week summer program that is Janelia-wide. It's a fun program. I also do take kids that want a gap year. So, I worked in industry for 4 years before going to graduate school. It was awesome. I highly recommend it. I've had 2 students who have spent a year or 2 in my lab before going to graduate school, and they're, at MIT and Caltech. So, I'm also happy to take resumes for that sort of thing.
Thank you.
Hi, my name's Emily. I'm wondering, with all these rings that are like the four-membered ring, whatever, do you have to store it under special conditions to keep it from reacting with the atmosphere?
That's a great question. So, the first thing we did, so my awesome chemist, John, made this molecule, came out of the HPLC. We were like, oh my gosh, this is amazing. It looked like it was Photoshopped, but it was real life. And so, then I went and I usually do the spectroscopy in my lab. Janelia group leaders are still expected to work in the lab. So I did the spectroscopy. We got this awesome quantum yield value of almost 90%. And so the first thing I did is we threw in a bunch of thiols, right? Because in a cell, there's a lot of glutathione and other free thiols. And we thought, oh, there's going to be problems here because you've got this positive charge. It's going to make this really susceptible to nucleophilic attack. And so we incubated it for thiols overnight, no change. We've had this stuff on the bench in DMSO at room temperature for a year, no change. So it's surprisingly stable, you know. But, you know, in hindsight, there are pharmaceuticals with, you know, where you would have a full positive charge. There are nicotine analogs, for example, with acetidine rings in them. And there you have a full positive charge, and those are able to be just sold normally. So in hindsight, the stability of acetidine maybe isn't so surprising. But yeah, we lucked out.
Any other questions?
So I have a question. This is Desmond Murray. So if you were to substitute instead of alkyl groups and cycloalkyl rings, aromatic groups, how does that change things?
Right. So in general, yeah. So in general, I'll just show you. So we've done that. And in general, these sorts of dyes are not fluorescent. And it's probably because now the ionization potential here is very low. There's been a couple recent reports of when you stain cells with these molecules, for some reason, they somehow are in some environment like the mitochondrial membrane, where they can become fluorescent, and still kind of not well understood why that is. But in general, for a technique called Forster resonance energy transfer, FRET, the dyes of choice as non-fluorescent FRET quenchers are N-aniline or N-aryl rhodamine dyes, actually. So yeah, they're strongly absorbing but not very fluorescent.
I think we got a couple more questions here.
So hello, my name's Noah. I think I remember you talking about taking images of the DNA or RNA traveling within the cell. Was that what you were saying beforehand?
Right. So we're actually looking at proteins usually. So in this case, this is a transcription factor. It's called SOX2. It's one of the main transcription factors that determines the fate of an embryonic stem cell. And so these are proteins that interact with DNA. There are methods, and we're working on methods in collaboration with other people to track RNA or to label DNA. But in general, the best labeling techniques are all sort of protein-based. Because we can encode them in the cells, we can tell the cell, hey, make this protein, and it will, and because we know so much about enzymes, we can actually have cool proteins being made. RNA and DNA are a little bit more difficult.
Thank you.
Hi, this is Dylan again. Can you go back to the slide where you were looking at the various rings and the quantum yields?
Yeah.
That one, yeah.
Yeah.
So I might have missed this in the video presentation. What I see is, yes, it improves the quantum yield, but at least in the other 3 ring systems, I did not see a huge increase in the brightness. So what prompted you to think that the 4-membered ring, I can understand why you would imagine a greater quantum yield, but what prompted you to think it would be brighter?
Right. So brightness is really how much the light the molecule absorbs,
right,
and the quantum yield, right? So we thought it would be brighter because we should get an improvement in quantum yield. We were surprised to also see a 25% increase in absorption. And so that was not expected, actually. So overall, we expected an increase in quantum yield. So you get twice as many photons out if you put the same amount of light in. Just based on quantum yield. But because the molecule also absorbs more light, that boosts you by another sort of 50%.
Yeah, okay, so I can see it. You were originally looking for frequency of photons. I just thought you were also looking for a higher amplitude with the 101, but okay.
Yeah, no, that was we've been trying to run some calculations to look at, you can do like oscillator strength to sort of predict absorptivity, and it does have a higher oscillator strength, but it doesn't really correlate well. For example, oddly enough, if you have ethyl groups here instead of methyl groups, you also see a large increase in absorptivity. And the 7-membered rings also show a large increase. So there's something funny going on with the cross-section of the dye, its interaction with light, that we don't understand.
I think I have one last question. Amazing talk and great background. Thank you for that. I wonder, how much do you worry about the imaging molecule interfering with the biology of what's going on? How much? I could see in some cases where it may not be much of an issue, but where does the molecule, the molecule, do you worry that much about it interfering with the biochemistry that you're trying to look at?
Yeah, no, it's a big problem, right? So right now, we hook, oops, I went too far. We hook a big protein onto sometimes not that big of a protein, and then we expect it to be the same. So, what we try and do is sort of due diligence to make sure that the cells are, grow the same rate, differentiate the same, et cetera. And we also do controls, making sure that it's not the halo tag that's interacting with the DNA or anything like that. So we can do our best, but really what's needed and what we don't have are small tags. As I mentioned, I put this number here, 34 kilodaltons, because I want to make sure everyone knows that this is a fairly large protein, right? So you have to be very careful. What we really need are methods where we can just put a small number of amino acids, or even look at endogenous proteins somehow and label them. And those have been actually quite difficult. You can have a small peptide and have another protein and then have that enzyme hook on the thing, but those are just harder to get to work. There's more moving parts. Right now, this is the best game in town, but maybe one of you guys will figure out a new way to label proteins and revolutionize everything.
Just one word, dendrimers. Yeah, it's just familiar. Yeah. Here. Okay, well, thank you very much. I think that's it for today. Let's thank our speaker.
All right, thank you, guys.
We really appreciate it. Maybe you'll get a few days off here to recover as the snow comes in.
Yeah. All right. Well, have a nice evening, you guys. Thanks so much. Being a great audience.
All right.
All right.